Hello everyone, I am Abousamra. I am from Stony Brook University, and today I am presenting the paper Weakly-Supervised Deep Stain Decomposition for Multiplex IHC Images. It is a collaboration between Stony Brook University and Cedars-Sinai Medical Center. Multiplex immunohistochemistry, or MIHC, is a method for simultaneously labeling multiple biomarkers in the same tissue section. MIHC is a useful tool for guiding immunotherapy. It allows to identify different types of immune cells in the tumor microenvironment and analyze multiple cell populations in terms of the population size, the spatial colocalization of different cell types, and the cell's shapes and sizes. It can be visualized by bright field light microscopy, making it a cost-effective solution compared to multiplex immunofluorescence, which is approximately 10 times more expensive. MIHC image capture is a single image with all the stains present together. This makes the analysis challenging, as a given cell may contain multiple biomarkers simultaneously. Different stains and their combinations may have significantly overlapping color spectra. In addition to the large variation in the expression of the same stain that is caused by varying amounts of the protein biomarker present, and the fuzzy appearance of the stains as shown in the samples below, all this makes it difficult for analysis. Color decomposition or color deconvolution is a useful analytical tool. It separates an image into its composition stains. The output is multi-channel concentration maps which represent the concentration of each of the basic colors in the image, that is, the stains and the background color. Here's an example H&E stained image and the resulting concentration maps. The purple is the hematoxylin staining the nuclei, the pink is the eosin staining of the cytoplasm, and last is the background color. In this paper, we address the color decomposition problem in MIHC images. We propose a scalable, deep learning-based color decomposition method. As far as we know, existing methods can decompose up to 4 color chromogenic stains images. Our proposed model can be trained to decompose many stains in the same image by incorporating weak annotation training data. Here's an example MIHC image and the resulting row concentration maps for 5 immune cell classes marked by yellow, teal, purple, black, and red stains. Annotating the stain masks is time-consuming and is practically impossible to get decent amount of training labels. We need to annotate in a time-efficient manner. Meanwhile, our network needs per-pixel supervision to train. We ask pathologists to dot approximate centers of cells. The dots are expanded to nearby area using superpixels. The resulting per-pixel label is not exactly accurate, but is a weak form of supervision. This is our proposed model, ColorAE or color autoencoder. The multiplex stained image is fed through the network. The output of the color autoencoder is the stain concentration maps. During training, the concentration maps are used to reconstruct the input image. The reconstructed image is compared to the input in the reconstruction loss. Additionally, the concentration maps are used to reconstruct the input using only the label stain at each pixel. This reconstruction is compared to the input in the label consistency loss. Next, we will explain the loss functions in more detail. We first explain how the image is reconstructed from the concentration maps and the basic stain colors. In our dataset, we have 7 basic colors. As shown by the figure, they are from 5 chromogenic stains marking 5 different immune cell types, in addition to hematoxylin staining nuclei in blue and the grayish tissue background color. We use the optical density to represent color. The optical density denotes the total loss of light when reflected by the material. It is expressed by the negative log of the RGB representation. For N stains, a pixel's optical density Y is a linear combination of the stains S1 through Sn in the optical density space and the stain concentrations at that pixel C1 through Cn, as shown by this equation here. Jointly solving all together, we have y equal s times c, where y is the image pixels, and s is the stain matrix in the optical density space, and c is the stain concentration maps. Our deep neural network will learn to predict the concentration map C hat. The reconstruction loss is the mean squared error between the input image and the image reconstructed from the predicted concentration maps. But the reconstruction loss is insufficient to train our model. When the number of stains is larger than 3, there are infinite solutions that satisfy the reconstruction loss function. Thus, we need additional supervision. The solution we are looking for is where at each pixel the label stain has the majority concentration. To that end, we propose the label consistency loss. We use the weak superpixel labels to reconstruct the image using the predicted concentrations of the label stains only. The label consistency loss is the mean squared error between the input image and this reconstruction, so that using only the label stain at each pixel, we get an image that is as close as possible to the input. To evaluate our model, we implement several baselines for comparison. ColorAE is a segmentation deep learning model based on the UNet architecture. It is trained with cross-entropy loss on the superpixel labels. Pseudo-inv or pseudo-inverse is a naive, unsupervised baseline. It solves the color decomposition equation y equal s times c using the pseudo-inverse of s. When there are many stains, we want to enforce that each pixel has one or few stains with notable concentrations, and so lasso-reg or lasso regression method proposes to solve it as a regression problem with L1 regularization to impose sparsity constraint on the concentration map. We show qualitative evaluation of the row concentration maps. Here are sample estimated stain concentrations for the 5 immune cell classes using our proposed model and the baseline models. ColorAE predicts poor concentration maps. They are the output from the softmax layer, and so they tend to be uniformly non-zero. Pseudo-inverse concentration maps have little sparsity, while lasso regression shows more sparsity. In both pseudo-inverse and lasso, different stains often share high concentrations in the same location, contradicting the visible stain as shown in the areas marked by the arrows. On the other hand, the concentration maps from ColorAE show higher quality. They exhibit both sparsity and that the concentration values are proportional with the visible stains in the image. We derive segmentation maps from the concentrations by assigning the stain with the maximal estimated concentration at each pixel. Here is the results from a sample patch. We can see the segmentation from lasso regression is better than the pseudo-inverse, but it is not as good as ColorAE or ColorAE. Both ColorAE and ColorAE are trained on the same weak labels, but our proposed model, ColorAE, is more sensitive to the stains. It better captures the stain boundaries and small components as shown in the circled regions. For quantitative evaluation, we calculate the F-score of the segmentation results. The ground truth is the dot annotation. A dot is counted as true positive if it intersects with a predicted component of the same label. Otherwise, it is counted as a false negative. And false positives are predicted components that do not intersect with any dot of the same label. The table shows the F-scores with ColorAE have the highest scores in almost all classes. We further validate our model using single stain slides. We have additional slides that are stained with only a single chromogenic stain and hematoxylin. Standard color deconvolution methods work reasonably well on 2 stain slides and can be used to generate ground truth concentration maps. We evaluate the estimated concentration maps from our model that is trained on the multiplex images using structural similarity and dye score. The table shows generally high scores in both metrics. In conclusion, in this work, we introduce weak supervision to the context of stain decomposition. We present a deep learning-based stain decomposition method for MIHC images. Our proposed method is scalable to a larger number of stains. In the future, we plan to explore a completely unsupervised model and use our models for large-scale analysis. Thank you for your attention.